In photosystem 1, pigments in the antenna complex are going to absorb photons just as we saw the pigment molecules do in photosystem 2. The energy from the sun is going to strike the antenna complex chlorophyll molecules, which will excite electrons, and then as the electrons drop back down to ground state, the energy will be passed from chlorophyll to chlorophyll to chlorophyll until it reaches the reaction center. Now, once it hits the reaction center, the electron in the reaction center will get excited, and this electron will be passed to a different molecule. Instead of pheophytin, it will be passed to ferredoxin. Now, if you remember in class, I told you that technically the electron that comes from the reaction center passes into the electron transport chain first, and then it reaches the ferredoxin. All I need you to understand, though, is that this time with photosystem 1, instead of pheophytin being the electron acceptor, ferredoxin is. So once the electrons get to ferredoxin, they are then passed to NADP reductase, which is an enzyme that ultimately reduces NADPH. Both NADP plus reductase and photosystem 1 are anchored to the thylakoid membrane. Remember, the electron chain proteins in general are anchored to their respective membranes. Ferredoxin is very closely associated with the membrane as well. So here in this image we can see a picture of photosystem 1. We see that same energy scale over here on the left, from lower energy at the bottom to higher energy at top. This is a measurement of the energy of the electrons. We can see photons coming in and hitting the chlorophyll molecules, exciting the electrons in the chlorophyll, which then pass their energy on to the next chlorophyll, until ultimately that energy hits the reaction center and causes the reaction center electrons to get excited. They're passed into the electron transport chain, where they're used to reduce ferredoxin, and then used to produce NADPH. So photosystem 1 produces NADPH as its product. Photosystem 2, remember, produces ATP as its product. You could think of these two product molecules as reducing power for NADPH and chemical energy for ATP. The question then is, how do the two photosystems connect with each other? How do they work? When we put the schematics for the two photosystems side by side, you see something that looks somewhat like the letter Z that's been turned on its side. And this is what we end up calling the Z scheme. It describes the way the two photosystems are linked. So if we follow from the beginning, we can see photons coming in from the sun, getting passed around the antenna complex, and ultimately exciting an electron in the reaction center in order to reduce pheophytin. Pheophytin then donates its electrons into the electron transport chain. They're carried around by a molecule called plastoquinone. Plastoquinone also generates a proton gradient, which flows through ATP synthase and helps to make ATP. Over here in photosystem 1, we have photons coming in and interacting with the pigment in the antenna complex, ultimately exciting electrons in the reaction center, which are then passed into the electron transport chain, reduce ferredoxin, and ultimately produce NADPH. Here's another reminder that we have to keep reducing the reaction center inside photosystem 2 if we want it to be able to continue to get excited. And those electrons come from the splitting of water. Water splits into oxygen plus hydrogen plus electrons. And the reaction center continues to have an electron available to get excited. The other thing I want you to notice is right here, because there's a molecule connecting photosystem 2 and photosystem 1. A couple other things to note. In photosystem 2, when the photons strike the chlorophyll in the antenna complex and get transmitted to the energy gets transmitted to the reaction center. It turns out what makes the reaction center unique functionally is that it's actually a special pair of chlorophyll molecules that we call the P680. Excited electrons from the P680 reaction center end up being passed to thiophytin, step down through the electron transport chain, carried by plastoquinone in order to create the proton gradient, which flows through ATP synthase and creates ATP. At the end of photosystem 2, though, we end up with electrons that need to go somewhere. They need to be used. They need to reduce an atom. They need to be gained or accepted by some molecule. So this is the molecule that links photosystem 2 and photosystem 1, and it's called plastocyanin. Plastocyanin. Now, this should not be confused with plastoquinone. Plastoquinone is the molecule that helps move electrons from protein to protein within the cytochrome complex, the electron transport chain. Plastocyanin receives electrons at the end of photosystem 2 and feeds them in to the beginning of photosystem 1. Plastocyanin is also capable of diffusing through the membrane, and it donates those electrons into photosystem 1 to the reaction center directly. And as we saw in photosystem 2, the reaction center in photosystem 1 is really a pair of chlorophyll molecules, and we call these P700. So plastocyanin essentially shuttles electrons between the photosystems. They're taken from photosystem 2 and passed over to photosystem 1. Now, ultimately, they're going to help reduce ferredoxin, which then passes them to NADP plus reductase, which then helps convert NADP plus to NADPH, which again is the product from photosystem 1. The electrons that are removed from the reaction center in photosystem 2 get replaced by the splitting of water, and the whole system continues to work. Electrons travel here, and then travel here, and then get transferred to here, and then travel here and get accepted by this molecule. We replace the electrons here with water, and then they travel here, and they travel here, and they travel here, and they travel here until they're accepted by NADPH. And then the electrons in the reaction center here are replaced with water, and the system continues. Now, I wanted to say a word about the two reaction centers and why we call them P680 and P700. The Z scheme, when it was first identified, helps explain something that we see in photosynthesis called the enhancement effect. 680 and 700 actually refer to wavelengths of light. 680 nanometers is right in the area of red light. And it turns out that photosystem 2 will proceed at its maximum rate when we shine a light of 680 nanometers onto it. When we shine purely red light at 680 nanometers onto photosystem 2, it will run at a maximum rate. However, photosystem 1 will only run at a reduced rate. Now, if you shine a red light, what we call far red, of 700 nanometers directly onto the thylakoid membrane, photosystem 1 will run at its maximum rate. Photosystem 2 will run at a reduced rate. In other words, these two wavelengths, 680 and 700, will run one of the photosystems maximally and the other one at a reduced rate. But when both wavelengths are shined together, as would happen when sunlight hits the photosystems, both photosystem 2 and photosystem 1 run at a maximal rate. So that brings us back to this very simple but very important diagram that we've looked at a couple of times now, this summary diagram for photosynthesis, where we take energy from the sun and we use it to produce carbohydrate. So far we've done the first half of this diagram. We've explained what happens in the light reactions, or the light-dependent reactions, or as your textbook says, the light-capturing reactions. In the light-capturing reactions, energy from sun is converted into chemical energy of ATP and reducing energy of NADPH. Water is split to produce oxygen and electrons to keep these light-capturing reactions moving forward. Now we're ready to take the ATP and the NADPH along with carbon dioxide gas and use them to create carbohydrate. This is accomplished in the Calvin cycle. Now, the Calvin cycle was named after Melvin Calvin, and Calvin was interested in understanding how carbon dioxide was converted into glucose. And so he used a special kind of experimental technique known as a pulse chase experiment in order to follow carbon dioxide inside the cell and see exactly how it was transformed into glucose. So your book does a nice job of explaining pulse chase, but generally what happens in the pulse chase technique is that some starting molecule, in this case carbon dioxide, is given a radioactive label. So instead of using ordinary carbon dioxide with ordinary carbon, Calvin used a radio-labeled isotope, or a radioactive isotope, carbon-14. So he used essentially radioactive carbon dioxide, and he put it into green algae in order to follow it inside the cell. When you take a radioactive molecule like carbon-14 and you put it into a cell, you can use that radioactivity to expose film, and you can literally watch this molecule as it moves from place to place to place inside the cell. And Calvin used this technique in order to figure out what happened to the carbon dioxide. Ultimately, he discovered that this carbon dioxide was added on to a second molecule, some mystery molecule, in order to produce a 3-carbon molecule called 3-phosphoglycerate. 2 3-phosphoglycerate molecules were produced from carbon dioxide. It was this mystery molecule, though, that Calvin needed to further understand. Interestingly enough, 3-phosphoglycerate is one of the intermediate molecules that we see in glycolysis during cellular respiration. So it turned out that this unknown molecule was a 5-carbon molecule. So when you take this 5-carbon molecule and you add it to the 1-carbon carbon dioxide, you can create 2 3-carbon molecules. So 5 carbons plus 1 carbon gives you 6 carbons, 2 3 carbons. The 5-carbon molecule was ultimately discovered to be a molecule called ribulose bisphosphate. Ribulose bisphosphate, or what we shorthand as RUBP. Now, it was eventually determined that there are 3 distinct phases involved in this set of reactions that converts carbon dioxide into glucose. And these 3 phases make up the Calvin cycle, or essentially the reduction of carbon dioxide into glucose. So I would like you to learn about these 3 phases. We're not going to learn all the individual intermediates that are involved in the Calvin cycle, at least not in this course. But I'd like you to understand what each of the 3 phases is about. Now, the phases are referred to as the fixation phase, the reduction phase, and the regeneration phase. What's happening in the first phase, the fixation phase, is that the carbon dioxide gas, which is coming in, is being attached to that 5-carbon molecule called RUBP. We're essentially fixing the carbon dioxide. We're making it into a more usable form by attaching it to this 5-carbon molecule. And ultimately this produces 2 molecules of 3-phosphoglycerate. The 3-phosphoglycerate is then fed into the second phase of the Calvin cycle, known as the reduction phase. And in this phase, we're going to use the 2 product molecules that were created during the light-dependent reactions. We're going to use the ATP that was produced by Photosystem 2 and the NADPH that was produced by Photosystem 1. So the 3-phosphoglycerate comes together with the ATP and the NADPH and creates a molecule called glyceraldehyde 3-phosphate, or what we shorthand as G3P. This G3P molecule is then fed into the regeneration phase. G3P is used for 2 different things. It's split up. Some of the G3P is used to create glucose and other carbohydrates like fructose. The remainder of the G3P is used to create more RUBP, which then is used during the fixation phase to fix more carbon dioxide and make more 3-phosphoglycerate, which comes together with ATP and NADPH to make glyceraldehyde 3-phosphate, which is split into 2 groups to make more carbohydrate and more RUBP, which goes back into the fixation phase and comes together with more carbon dioxide. And now you understand why we call this the Calvin cycle. It's a cyclic reaction. It continues on as long as we have RUBP available. It's similar to the Krebs cycle in that unless there's oxaloacetate available, we can't feed acetyl-CoA in. So we have fixation, reduction, and regeneration, and those 3 phases make up the Calvin cycle. Make a note here that all 3 phases of the Calvin cycle are taking place in the stroma. That's the space in the chloroplast that's in between the outer membrane and the inner thylakoid membrane. So once again, RUBP comes together with carbon dioxide to make 3-phosphoglycerate. The 3-phosphoglycerate then comes together with ATP and NADPH to make G3P. Some of that G3P is used to regenerate the RUBP, and some of it is used to make carbohydrate. Now we often draw the Calvin cycle, as a clock face, in order to remind us that this is a cyclical reaction. It just keeps going on as long as we have carbon dioxide and RUBP available. So this 1/3 of the pie refers to fixation, where RUBP combines with carbon dioxide to make 3-phosphoglycerate. And I like this diagram because this diagram reminds us that RUBP has 5 red carbons and carbon dioxide has 1. And when they come together, we get 2 molecules of a 3-carbon molecule called 3-phosphoglycerate. This 3-phosphoglycerate is fed into the second stage, the reduction stage. ATP gets hydrolyzed, NADPH gets oxidized, and we create this molecule, G3P. Again, a 3-carbon molecule. Some of the G3P feeds out to create glucose and fructose and other carbohydrates, and the rest of it regenerates RUBP. Notice that we have to hydrolyze a little more ATP here in order to regenerate the RUBP. So we hydrolyze ATP, we oxidize NADPH during reduction, and we hydrolyze a little more ATP during regeneration. So the Calvin cycle helps us understand what happens to the products of the light-dependent reactions. The Calvin cycle explains where the ATP and the NADPH go, where those products of the light reactions are ultimately used, and used by the cells to reduce carbon dioxide. One of the lingering questions that came out of the early work by Calvin was the question about this reaction between carbon dioxide and RUBP. It turns out that adding carbon dioxide to this unusual 5-carbon molecule in order to ultimately create some carbohydrate, this is one of the 2 reactions that's unique to the Calvin cycle. In other words, we don't see this occurring anywhere else. And of course, this is an incredibly important process because it's through photosynthesis that we create the carbohydrate that's used by the organism to do cellular respiration. The plant has to make glucose and other sugar in order for organisms to be able to burn that sugar, oxidize that sugar, and perform cellular respiration. So one of the last questions that Calvin and others had was, what enzyme catalyzes this reaction? What enzyme helps to bring together carbon dioxide and RUBP and create 3-phosphoglycerate? And it turns out that this enzyme is really quite interesting, which is why we'll learn just a little bit about it today. This enzyme has a terrible name. It's called ribulose 1,3-bisphosphate. We shorthand that name into RUBISCO. So the real name is ribulose 1,5-bisphosphate, and the shorthanded name is RUBISCO. So this is an enzyme, and this is an enzyme that's found in all photosynthetic plants. And what makes it so fascinating is that it's thought to be the most abundant enzyme on Earth. Remember, this is the enzyme that fixes carbon dioxide and RUBP and produces 3-phosphoglycerate in order to produce carbohydrate. This is the enzyme that produces the vast bulk of the carbohydrate that is present on the planet. The carbohydrate that all of us heterotrophs, those of us who can't do photosynthesis, the carbohydrate that we rely on as a food source to do cellular respiration. What's particularly fascinating about RUBISCO to me is that it's shaped like a cube. Its tertiary shape is literally something like a cube. It has 8 faces, like a cube does, and each face has an active site on it. So RUBISCO has 8 active sites. The other thing that's interesting about RUBISCO is that it's particularly slow. It's thought to be a very ancient enzyme, and it's only capable of catalyzing about 3 reactions per second. And if you remember, some of the enzymes we've talked about in our course have been able to do as many as 50 reactions per second or 500 reactions per second. So this is a relatively slow enzyme in its ability to perform catalysis. It turns out that RUBISCO does 2 separate reactions. RUBISCO does the reaction of catalyzing carbon dioxide coming together with RUBP, what we've just talked about in photosynthesis. It's also able to add oxygen onto RUBP in a process called photorespiration, which is done by certain organisms on the planet. So RUBISCO is quite unique. This last slide I wanted to show you just puts together some of that information about RUBISCO for us. The RUBISCO is capable of doing 2 reactions, both photosynthesis and photorespiration. And if you remember when we talked about enzymes, this is very unique. Most enzymes are very good at doing one thing. Most enzymes perform only one reaction. And here we have an enzyme that's capable of 2 very different reactions. In photosynthesis, carbon dioxide comes together with RUBP to create 3-phosphoglycerate for the Calvin cycle. But in organisms that can do photorespiration, RUBP is placed with oxygen to create 2 intermediates, 3-phosphoglycerate and 2-phosphoglycolate. The 3-phosphoglycerate is used in the Calvin cycle, and the 2-phosphoglycolate is diverted for other reactions. Now, I don't need you to learn these intermediates. I don't need you to have a deep understanding of photorespiration. I just want you to understand that the RUBISCO enzyme is quite unique because it's not only is it a particularly abundant enzyme, the most abundant enzyme on the planet, it's also very unusually shaped like a cube. It has 8 active sites. It works very slowly, relatively speaking, but it's capable of performing 2 completely different reactions. Really quite unusual enzyme.